Hello, my name is Manuel Avila, and I'll be presenting APOE gene associated with cholesterol-related traits in the Hispanic population. Several genes have been suggested for abnormal lipid or lipid-related phenotypes. Among these candidate genes, the apolipoprotein E gene is well documented. APOE is a multifunctional protein which plays an important role in lipoprotein metabolism and has been shown to be associated with maintaining homeostasis of cholesterol levels and transporting lipids. The genetic polymorphisms of the APOE gene is mainly attributable to three alleles: epsilon 2, epsilon 3, and epsilon 4. The APOE epsilon 4 allele is associated with lipid-related traits or diseases mainly reported in non-Hispanic populations, which had never been explored in the Hispanic population. Thus, the current study investigated the frequencies of the APOE alleles and studied their relationship with cholesterol levels and lipid-related traits in the Hispanic population. Material and methods. This study is an analysis of data collected by the Texas Alzheimer's Research and Care Consortium (TARC), 1,320 subjects, in combination with your data initial study of longevity and dementia from the real around the body, 62 subjects, resulting in a total of 1,382 subjects. DNA isolation and genotyping. Genetic data were collected to study genetic determinants such as the APOE genotypes and alleles. DNA extraction was performed using two different methods. 1,320 blood samples were collected from TARC, and 62 soluble samples were collected from the RGB study. The genotype for the APOE gene was determined by two separate methods. Taqman single nucleotide polymorphism assay were used to analyze the SNPs, and RS74112 and RS429358 were used to identify the three main APOE alleles for the RGB study sample. TARC utilized the Affymetrix Genome 1 Human SNP array 6.0 to collect the SNPs RS74112 and RS429358 data. Statistical Analysis. Statistical analysis was performed according to the APOE epsilon 2, epsilon 3, and epsilon 4 carriers and non-carriers. In total, several variables were used to generate the statistical reports from the study population. The statistical package for social science SPSS version 26 was used to perform the statistical analysis. Power analysis. Power analysis was estimated for known genes using a case control study designed for discrete rates using genetic power calculator. Results and discussion. According to Table 1, a total of 1,320 out of 1,382 participants had APOE genotype data. The status of the 1,320 participants was classified as 860, 65.2% participants with high cholesterol levels, while 460, 34.8% represented the control group in the present study. A total of 589 females and 271 males had hypercholesterolemia. The mean age of hypercholesterolemia cases was 70.49 plus 9.12 years, which was found to be statistically significantly older than in the control. The frequencies of the hypertension, thyroid diseases, and diabetes were statistically significantly higher in the patients with hypercholesterolemia as compared with the control group. P was less than 0.002. There was no difference in body mass index between hypercholesterolemia and the control group. Distribution of the APOE alleles. A total of 961, 69.8% subjects were females, and 421, 30.2% subjects were males. The allele frequencies of the APOE epsilon 3, 96.7%, epsilon 4, 22.6%, and epsilon 2 6.8% alleles were observed in Table 2, which are similar to the frequencies in the Hispanic population in previous reports. APOE epsilon 4 allele status and demographics and cholesterol-related phenotypes is displayed in Table 3. In terms of the APOE allele epsilon 4, a total of 20.6% females and 22% males carry at least one APOE epsilon 4 allele. A total of 325.9% participants with hypercholesterolemia carried at least one copy of the APOE epsilon 4 allele. Chi-square tests revealed a significant association between APOE epsilon 4 allele and hypercholesterolemia. P was equal to 1.17 times to the minus 4. Multivariable logistic analysis. A multivariable logistic regression analysis was performed, and the results further support the association between the APOE epsilon 4 allele and the hypercholesterolemia. According to the Table 4, after controlling for potential confounding factors, example gender, education, and APOE epsilon allele, we observed a statistically significant association between APOE epsilon 4 allele and hypercholesterolemia. P was equal to 1.8 times 10 to the minus 4. The odds of having hypercholesterolemia were 1.9-fold higher in persons with an APOE epsilon 4 allele compared with those without. The APOE epsilon 3 allele was not associated with hypercholesterolemia. P was equal to 0.484. Conclusion. The current study emphasizes the prevalence of high cholesterol levels and the effects that this may have on genetics and demographics, example, sex, age, and education in the Hispanic population. The results of the current study demonstrates that the APOE epsilon 4 allele carries a high risk for cholesterol-related traits, including hypercholesterolemia in the Hispanic population. No associations were found between the APOE epsilon 3 or APOE epsilon 2 allele and cholesterol-related traits in the current population. While this study aims to aid doctors in the provision of future care plans to detect and prevent metabolic syndrome symptoms, further studies are needed to confirm our current finding in this population. This work functions to highlight how the understudied Hispanic population can further our understanding of the genetics of cholesterol